to be here today and to have the opportunity to share with you some of the work we have been doing in the lab of Oscar for the past few years, and also have the opportunity to discuss these matters directly with Professor Song Hai. I think it's a very nice opportunity for both of us to realize which are our main points of discrepancy, which I always wondered, how much of that is based on semantics or how much of that is based on real scientific discrepancy. But in any case, as you said, our work has been focused in understanding the developmental principles and developmental mechanisms that account for the generation of excitatory cell diversity in the cerebral cortex. So as Professor Hai elegantly introduced too, the cerebral cortex of mammals is made of different populations of neurons that can be classified based on a combination of multiple structural and functional criteria. The most prominent neuronal class in the cerebral cortex are excitatory projection neurons, also known as pyramidal cells. These cells occupy the different laminar, different layers in which the cortex can be divided in the radial dimension, with the exception of layer 1, and also presenting different populations of progenitor cells within each of these different, of pyramidal cells within each of these different layers. In this big cell class can be further split into different subclasses. It has been classically classified into two main subclasses: intertelencephalic projection neurons that occupy all different layers of the cortex and project to a number of targets inside the telencephalic territory, and corticofugal projection neurons that mainly populate the deepest layers of the cortex and project to diverse structures outside the telencephalon. Now, of course, this is just at the surface of the diversity of excitatory cells, since many different types can be subdivided from these subclasses based on their precise projection patterns and the expression of certain genes. And recent RNA-seq data seems to be confirming that this diversity is way deeper than this, with a few dozens of cell types have been identified in different regions of the cortex. They seem to be also having remarkably important differences in different regions of the cortex in terms of cell diversity. Now, how this diversity arises from development was also very nicely introduced by Professor Song Hai, is relatively well understood. We know that during early development in the presentive cortex, population of stem cells named neuroepithelial cells divide symmetrically to expand the progenitor pools and to generate these radioglial cells, which are the main progenitor cells in charge for the generation of excitatory neurons. These cells began the generation of neurons around the embryonic day 12 or so in the mouse, and they do generate these excitatory cells in two main ways, either as a direct result of their mitotic divisions or through the generation of intermediate progenitor cells, which are transient amplifying progenitors that divide typically once to generate two postmitotic neurons. Excitatory neurons born in these two ways then get attached to the basal process of these radioglial cells and use those as rails to migrate radially and invade the growing cortical plate. And this process is very highly temporally organized. So the earliest cell derived from the earliest division of radioglial cells will occupy the deepest strata of the cortex. And then later generated cells will migrate above previously allocated neurons and occupy more superficial positions, differentiating also into more superficial fates. Now, although it's collectively well accepted that this population of radioglial cells is responsible for the generation of this entire diversity of excitatory neurons, how individual radioglial cells do this job has been a matter of recent debate. The most accepted and the most classic model that also Song Hai was proposing in his previous talk basically postulates that individual radioglial cells do transit through a number of competence states, which are illustrated here with these colored squares. And in each of these competent states, they will be competent to generate a specific type of excitatory neuron. Therefore, the transition of individual radioglial cells across these different competence windows, along with developmental time, will allow them to generate the entire set of pyramidal cell types that populate the adult mammalian cortex. Now, other studies have proposed alternative models for cortical neurogenesis in which the generation of this set of cell types will be coming from the collective effort of different pools of progenitor cells that will be somehow pre-specified to generate certain types of excitatory neurons. Now, when studying these outputs of these individual progenitor cells, something that should be taken into consideration is the fact that the cerebral cortex is not an homogeneous structure. That is indeed made of different regions, different areas, which know different functions, and that also present remarkably different cellular architectures. This can be noticed in the human cortex from the drawings of Kehal, but it's also very clear in the mouse cortex. So different areas of the neocortex of mammals present different architectures with different fractions of cells or the different lamina identities. And although we know that during early development, this segregation of different regions relies in the activity of certain patterning cues that generate a protomap that will end up into this segregation, how these signals do impact the activity of progenitor cells to allow them to generate these different cellular architectures, it's remained to be explored. So what we did in our study was very similar to what Professor Songhai was showing before. We mapped the output of individual radioglia cells and studied the composition of these neuronal outputs at adult stages, at P21, postnatal day 21, when the generation of cells and also the cell death that is associated with early postnatal life is largely finished. Now, previous studies have done this kind of work and have often led to conflicting results. In our view, one possible reason for this is the fact that all available methods to do this single cell fate mapping present different caveats and limitations that may compromise the interpretation of the results. So to circumvent the limitations of individual approaches, we use 3 different methods to do these individual outputs of progenitor cells, so that despite all of them present different limitations, the combined use of those can provide a better understanding of the process we are studying. So the first of this data set was generated by a former member of the lab, Gabriele Ciceri, who was injecting conditional retroviral cassettes at low titer and encoding for fluorescent reporters into the lateral ventricle of mouse embryos, in which Cre recombinase expression is confined in excitatory cells. These injections were done at T12.5, so at the moment these radioglia cells began the generation of neurons, and were able to target a very sparse population of progenitor cells and map their neuronal outputs. We will then analyze the brains at postnatal stages and study the composition of these outputs. So what we observe in these preparations are isolated clusters of excitatory cells that reflect the output of individual progenitor cells, and is what we denominate lineages. Here you have an example of 3 consecutive brain slices in which cells belonging to this lineage have been marked, followed by schematics in which we are collapsing all these cells into a single diagram for clarity. So the majority of lineages observed in this dataset were like the one you're observing in your screen, where what we call translaminar, that is composed of cells of different laminar identities. But we also observe a fraction of lineages that were composed exclusively of either deep or superficial layer neuronal identities. As I said, translaminar lineages were the majority, but the restricted lineages, either deep or superficial, also represent a visible fraction of all lineages observed. We also check the clonal size of those lineages, that is the number of cells that are composing these individual lineages, and observe that translaminar lineages were typically larger in size compared to restricted ones. Now, as I said, all these techniques present different caveats and different limitations, so we wanted to contrast this with other methods in order to see whether some of these observations were indeed artifacts of the way we are labeling these progenitor cells. Here we use same approach introduced before by Professor Songhai, mosaic analysis with double markers. We did this in collaboration with the Hippenmeyer Lab, which are great experts in the use of this technology. And here we paired this MADAM approach with mouse genetics to restrict the labeling in cortical radioglia cells, and we timed them, the triggering of this labeling at T12.5 using tamoxifen injections. Now, in this dataset, again, the majority of lineages were translaminar. Indeed, this majority was enlarged compared with the previous dataset. And we also observe a small fraction of lineages composed exclusively of deep layer fates. But it was very rare in this dataset to observe those lineages composed exclusively of superficial layer identities. Indeed, we consider those to be virtually absent in this dataset. Now, there are many possible alternative explanations for this discrepancy between the 2 datasets, given the technicalities of both techniques. But to make a long story short, MADAM mainly labels cells that are actively dividing at the moment we trigger the labeling with tamoxifen. Therefore, if we were to have a population of progenitor cells that won't be dividing early on, that will be quiescent early on during development at the moment we inject the tamoxifen, but only start dividing later on when superficial layers are mainly produced to generate the superficial layer restricted lineages, MADAM will very hardly pick such population of progenitor cells, but the retrovirus could pick this kind of population. So to test for that hypothesis, we generated a third data set in which we basically replicate the same experimental paradigm using our MADAM experiments, but substituting the MADAM reporters with a regular inducible GFP reporter, so that we will label progenitor cells irrespectively of their mitotic activity. And in this final data set, we were able to recover a fraction of lineages that was about 10% of all of them that was composed exclusively of superficial layer fades. This fraction was smaller to the one observed in the retroviral data sets, indicating that some of these observed lineages were artifactual and had to do with the technicalities of retroviral data. So all these methods together, what we were able to conclude here is that in the developing cortex of mouse, about 80% of ready glia cells do generate translaminar lineages, while about 10% of those generate deep restricted progenies, and another 10% seems to be not actively dividing during early embryonic stages, but start dividing later on to generate superficial layer restricted progenies. Now, as I said before, it is very important to note here that this doesn't mean these progenitors are somehow pre-programmed to do this kind of outputs. This can be alternatively a result of the regulation of their neurogenic behavior, as I will discuss in the subsequent slides. But first, to check a bit more in detail the structure of these lineages, we could do that and gain a bit more knowledge about what these ready glia cells are doing during development. So first of all, we observe that the clonal size distribution of these lineages was very variable from one ready glia cell to another. That is, the total amount of cells that a single ready glia cell produces during neurogenesis can go from 3 to over 10 cells within the same temporal window of time. So this is very variable, and this wide distribution of clonal sizes was true for both translaminar and restricted lineages, although restricted lineages, as I said before, were typically smaller in average size, and therefore did contribute largely to this enlarged fraction of lineages composed by only 3 or 4 cells. Now, this diversity in outputs not only has to do with the amount of cells these ready glia cells produce, but also in the cell identities they generate. Here you have 3 examples of translaminar lineages in which ready glia cells have skipped the generation of certain laminar types of excitatory neurons. Indeed, if we make a formal quantification of these configurations, that is, the configuration of the lineages based on the presence or absence of cells of the different laminar identities, we can observe that all possible configurations were seen in our dataset at a given frequency that is indicated in the bars below. So it seems that ready glia cells do many different things in terms of how many cells they generate and which laminar identities they generate. But we also wanted to check the composition of these lineages in projection types, and we did that using known molecular markers that have been classically used to identify those projection types. So here I'm showing the composition of lineages in projection types only for those lineages that are complete in laminar composition, so those that contain cells of all the different laminar identities. And as you can see also, lineages contain different combinations of these projection types, being quite rare, those that are composed by all different projection types and all different laminar types. So taken together, this data indicates that the neuronal output of individual ready glia cells seems to be extremely heterogeneous in both the number and the cell identities they generate over development. Now, how do we make sense of this diversity of outputs? One can think in many possible models. Here I'm going to be presenting probably the two most extreme models one can conceive, but of course, intermediate points between these two extreme models should be also considered. So first model, model A, will suggest that we need an equivalent level of heterogeneity at progenitor stage. That is, we need many different pools of progenitor cells that are somehow pre-specified to generate each of these different lineage compositions, each of these different outputs. On the other hand, a perhaps more parsimonious model would propose that a unique homogeneous population of progenitor cells could be able to generate all this diversity of outputs if the rules instructing their behavior were not to be fixed and deterministic, but rather flexible and stochastic. And when thinking in this model, one should conceive and one should consider the context in which red glia cells are operating during development. So these cells are under the influence of many different extrinsic signals that are known to modulate different aspects of their neurogenic behavior. So these signals are known to modulate the cell cycle speed of these cells, which can be differently modulated during different stages in their molecular progression. They can also modulate the intermediate progenitor cell versus neuron balance at these different points during their molecular progression, and also the length of the different competence states that these red glia cells transit through. And therefore, this will have a massive impact in the final output of these individual red glia cells. So these signals could be orchestrating the collective output of these cells by having differential effects of individual red glia cells, making them behave in a rather stochastic way, producing different and unpredictable outputs while controlling that they build an invariant structure, as is the cerebral cortex. So to test for the feasibility of this second option, to test whether such stochastic turbines can build effectively the entire diversity of configurations that we are observing in our experiments, we simulate progenitor cell behavior in silico using simple stochastic principles, and then compare the lineage structure output by these simulations with those observed in our experiments to see whether similar level of complexity could be produced with these stochastic models. So here, progenitor cell behavior is modulated following two basic principles. First, progenitor cell will have a number of chances to generate an individual excitatory cells within each of the different layers of the cortex, and this basically represents the temporal window of opportunity that a given in vivo red glia cell will have to generate cells of that particular laminar identity. And second, we will input a probability value that will be used to stochastically decide within each of these chances whether an individual pyramidal cell is generated or not. And these probability values somehow reflect or represent this entire set of different signals that can modulate different aspects of the behavior of the red glia cells. So these parameters will be fit for these progenitor cells to reproduce the collective output of those observed in our experiments, that is the correct laminar structure among all of them, and also to reproduce the clonal size distribution observed in our experiments. So progenitor cells that produce similar lineages in size, and that also respect the collective structure of the cerebral cortex. And then compare those with the configurations observed in our experiments to see whether they can produce comparable organizations in terms of laminar occupancy. And to our surprise, it turned to be that with a unique and homogeneous population of progenitor cells, just a single set of stochastic rules, we were able to reproduce the vast majority of lineage configurations we have observed in our experiments at approximate numbers, indicating that simple model based on unique homogeneous populations of progenitor cells and operating through stochastic principles are able to account for the majority of the configurations we have observed. However, these models seem to be not reproducing certain specific points in our data. So they were underestimating the fraction of lineages composed of superficial layer identities exclusively. And they were also producing a unimodal clonal size distribution that was peaked around the same size as those observed in our experiments, but failed to reproduce this enlarged fraction of lineages with small sizes that was linked to these restricted configurations in our in vivo dataset. So we generated a second version of the model in which these rules were kept the same for the majority of lineages, but we introduced a second minor population of progenitor cells that was tuned to preferentially generate superficial layer identities and also to preferentially generate smaller lineages. And here I need to stress that these progenitor cells were not restricted in fate. So they were allowed to produce the deep layer identities, but they were biased to produce more preferentially those of superficial layer identities. That's why, I'll need to take very careful with the conclusions we are making, because this doesn't validate, so this doesn't imply that this progenitor cell, this second progenitor population, will be restricted to produce exclusively superficial layer identities. So with this introduction of a second population of progenitor cells that was biased for producing superficial layers, we were able to replicate the fraction of cells, the fraction of lineages composed of superficial layer cell identities exclusively, as well as this bimodal distribution of clonal sizes that we have observed in our experiments with this enlarged fraction of lineages composed by only 3 or 4 cells. So this seems to indicate that the majority of the diversity observed in our experiments can be reproduced with models including a single progenitor population, and the few points that seem not to be satisfied by that can be satisfied by introducing a second progenitor population that is biased but not restricted to produce superficial layer identities. No one can think what's the use of such a mechanism. Why would the cortex benefit of having stochastic mechanisms in order to be built? I will then come back to my earlier point during my introduction. So different regions of the cortex are made of different cellular architectures. Here you have an example of primary somatosensory and primary visual cortex, in which excitatory cells have been labeled with a nuclear marker. So you can appreciate the differences in the ratios of cells or the different laminar identities, how they vary from one region to another. Then we use our models to check that with subtle tuning of the probability values that we implemented in those, we were able to reproduce the laminar fractions of cells that define these different cortical regions. So the collective output of progenitor cells in our simulations, given this subtle tuning, was able to be changeable pretty easily to adapt to those that define the different regions of the cortex. So it seems that this kind of stochastic model could account also for the adaptation of the progenitor cell behavior in order to build different cellular architectures as those that define the different areas of the cortex. So this is just my summary slide. So what we have been proposing today is that what we have been observing in our experiments is that the output of individual retiglia cells seems to be extremely heterogeneous in both the number and the cell identities they produce. But this not necessarily require the existence of different progenitor pools that are somehow engineered to produce this kind of outputs. Instead, these progenitor cells could follow the same or a very similar hardwired molecular pathway that will allow them to transit across different competence states, but then will be plugged to a population level modulation mechanisms that can make them take a number of stochastic decisions along their way, so that their final output will be very different from one cell to another without requiring the use of specific different hardwired programs for that. So then that will be all from me today. Just before finishing, we'd like to thank first my supervisor, Oscar Marin, for helping with this work. Also, all the Marin and Rico Lab members for helpful discussion and direct or indirect contribution to this work, and also my direct collaborations that were having enormous impact in the developing of this project. And of course, thank you all for your attention also.
Thank you, Alfredo, so much. It was a really, really cool talk. I really love your schematics explaining really complex ideas. They're really well done. So I'd like to open the floor for questions. And again, you can send them through the chat or pop your hand up in the in your Zoom video.
Yeah.
Alfredo, perhaps I can start with a prompt for you to continue that discussion that you had with Songhai early in his talk, and we are interested to hear your comment when you were addressing him during his seminar. Alfredo, could you articulate that now for the benefit of the audience? Thank you.
You mean about our discrepancies in how we conceive this?
Well, I think it's very interesting to the audience. Not all of us are working in lineage tracing work. That the data that's generated from both labs are equally valid for us to resolve what still is very mysterious about the generation of neurodiversity within the cerebral cortex progenitor cells. And so if one can bring closer both ideas that are supported by data and the approaches that they take, that would be really helpful here. And so could you make a comment on that? And Songhai, I invite you to also make a relevant comment there as well. And then following from that, perhaps we can, Kelly can raise Stefan's question that's in the chat box as well. Thank you. Yeah.
Yeah, so thank you for the question. So my bit on it, I don't think we are, I think our data pretty much converge in certain points. So for instance, we both see this distribution of clonal sizes within the MADAM clones, and we also see that with other techniques. So I think the data agrees on that. I think our, again, I don't know to what extent our discrepancy is semantics, but I guess Professor Songhai's conclusion out of that is that this is a unitary output, but we also, we kind of capitalize in the variability. So it depends on how you, Professor, define this unitary output and this deterministic behavior. I don't know how easy it is then to explain this variability. So for me, the point is that it seems that what a ready glial cell does is very different from one another. And I think that makes sense if you consider that these are regulated through many different signals. So of course, if you regulate a population of cells with extrinsic signals, you're definitely going to have to see that these output different things, right? So again, also Professor Songhai never said that, or he did state before that the composition of these lineages can be variable. So I think he also accepts that fact. Is that correct?
Well, I think yes. I think basically there are two issues, right? One is the size, one is laminar distribution. I think in terms of the size, I still think even in your study there is a constraint. The total output of the clone size. So this, to some degree, reflects there is a defined output of radial clear progenitor cell in terms of number of neurons. Then, in terms of the laminar distribution, clearly not every clone is the same. In fact, hardly you find any two clones that exactly have the same identity.
Exactly. Yeah.
So, but I like to think that this overall progressive from deep layer to superficial layer, that I think what Alfredo emphasized, there's a skipping of layers. Actually, there's one suggestions or questions I have is that if you take into consideration of the intermediate progenitor cell, which will modulate the output burst state of particular laminar corresponding time, that can lead to variation in terms of laminar output. So that does not mean there is no programmed neurogenesis process that in the, but on the other hand, it also means not probably some of the radial clear progenitor cell do not go through every round of division and go produce an intermediate progenitor cell. So I think there's not more for us to learn. I fully agree that actually, if you look into the essence of the data, actually there's not more. I think essentially the same data set you reproduced of the MADAM data set we analyzed before. It just, you look at from different perspectives. For example, in your study, I assume you focus more on those 20% of the clones that was more restricted, smaller size and superficial deep layer restricted clone. If you look at the vast majority of the 80% of the clone that is more translaminar, that have a much more organized size output. So I shouldn't take much more time. We can discuss more.
Sure, of course. No, elaborating on that. So, of course, we don't mean that there is no program underlying this. Indeed, we did some experiments, some models in which we just permute cell identities across lineages, basically based in the hypothesis that perhaps there is no program at all, and then you just need to produce certain amount of cells, and you can just randomly distribute these cells among lineages and you will observe something similar to what you're observing in vivo. That's not the case. So we do believe there is a program. The main difference is we do think that this program can be modulated. So you can, for instance, make a progenitor cell skip the generation of a layer 5 cells, despite they will have a window for doing this. You just have to, for instance, stop them from dividing over this time or so. And now again, of course, we do focus earlier in our publication, also in my presentation, in these restricted lineages because of the controversy they have been surrounded by. But at this point, I do not think these configurations are more relevant than those that, for instance, skip layer 5 or layer 4, right? They're reflecting probably the same kind of thing. Now, about the superficial layers specifically, our models seem to indicate you need different rules to match with the fraction of those. That doesn't mean that the general models, the stochastic models, cannot produce those. They do, but in smaller fractions compared to what we are observing in our experiments. So that seems to be that you need something else there to produce these configurations. But as I said, this doesn't necessarily need to be a restricted progenitor cell. So yes, I think generally, again, translaminar are more abundant. It's just that the laminar composition is really variable from one another, and that I think it really makes sense if you consider that these progenitor cells are not isolated in the vacuum, but exposed to regulatory cues.
Just a quick short comment.
Sorry.
1 minute. So in fact, when we hear Alfredo says that this is stochastic, actually this stochastic is actually very much constrained, at least from simulation, to fit with their data set. Each progenitor cell population takes about 9 parameters to constrain it to fit with the data set. So it's not really just a random process. I'd like to just to point to you. That,
no, of course. That's why our models are structured in a way. So they create layers and they have chances of generating cells of different layers. It's not a random process, of course. It's not completely random and cells generate identities, out of the blue. Of course, that's why I think, this earlier literature supporting this progressive transition across competence states, it should be considered, and of course we consider that that's the case. It's just that the amount of cells you generate of the different identities is very variable, and that requires something else than this progressive transition across competence states.
That's great, Alfredo. I think we're going to have to stop it there. I really love the